Hello, everybody. Welcome back to Creation Myths. Today, I am thrilled to have with me Dr. Herman Mays, and we are going to be talking about Traced by Dr. Nathaniel Jeanson. Before we go any further, I'm just going to make sure that I can hear myself okay on YouTube, and it sounds good. So we're good to go. As always, if anybody has any audio problems, just let me know in the chat, and I'll see it, and we can deal with it. So today's show is a lot of fun. It's kind of a follow-up to my review of Traced from a couple weeks ago. My guest today is Dr. Herman Mays. He has a PhD in evolutionary ecology from the University of Kentucky, and he is an associate professor in genetics at Marshall University. Dr. Mays, thank you so much for coming on. Anything else you want to say to introduce yourself?
I mean, that pretty much covers it, I guess. I have a day job as a scientist in biology. We do molecular systematics and population genetics, mostly in birds. But I've had this sort of lifelong interest in science denialism and conspiracy theories and this kind of thing. So that's what leads me to read all these creationist books.
Just constantly getting sucked back into the creationist circle.
Yeah, like we talked about before, is kind of somebody's got to do it. If you from them doesn't mean they go away. So
yeah,
it's good to let people know what this is and what it isn't.
Exactly. So today I'm really excited for this because you are kind of like one of the Nathaniel Jeanson people. In terms of just dealing with the claims made by Dr. Nathaniel Jeanson from AIG. And today we are talking about his 2022 book called Traced. So I have reviewed Traced. That link is down below in the description. Dr. Mays has also reviewed Traced in text form on his blog. That link is down below. And he also wrote up a more detailed look at some of Jeanson's methodology, just really digging into exactly what's going on kind of behind the scenes in terms of how Jeanson does the calculations in his book. And that's also, a link to that is also down below. So definitely check that out. If you want the more 30,000 foot view, check out the general reviews from each of us. But today we're really going to focus on the specifics of the methodology and Traced and really kind of follow Jeanson's data down the rabbit hole and see where it turns up. So I don't have anything more by way of introduction other than to say that Dr. Mays is kind of famous in the Jeansonosphere for having debated him several years ago on replacing Darwin on the Non Sequitur show, which is one of the few public debates that Jeanson has ever done on his work. So if you haven't watched that, I encourage you to hop onto YouTube and check that out. So, Dr. Mays, before we get started, I have just one simple question for you, and then we can get into the more specifics. Have you read the book?
Yeah.
Just wanted to make sure because that was a point of contention in the past.
I have it here.
There it is.
And, a dog-eared copy of it that you can see.
There you go. Yeah.
And I avoided the mistake this time. The first book, I bought the Kindle edition. And I'm so glad I didn't do that with this one because the Kindle edition, if anybody's interested in reading this, even positive reviews of it on Amazon say the Kindle edition is impossible to read.
Yeah.
Just because of the format of it. But
guilty.
Yeah.
The way I ended up doing it is I would kind of two-screen it where I would have on my desktop, I'd have the figures open while I was reading on my iPad on the Kindle app, be reading the book, but I'd have the figures open on my screen. So I didn't have to constantly go back and forth because why you would be constantly referencing figures in a text and not have the figures exactly below the paragraph where you reference it is beyond me.
Yeah. I mean, it was just madness. So I definitely read it. It was easier to read this time on a paper version than the first book was. That doesn't mean I enjoyed it anymore, but yeah.
So we could be here all day if we wanted. I have an itemized list of the problems, big and small, with this book, but I really want to focus on what you wrote about in that second post that you put on your blog that's linked down below, really digging into Jeanson's methodology in terms of how he calculates his mutation rates. So for anybody not familiar with Jeanson's work, kind of like the thing he does is he uses a strict per generation molecular clock, just 3 mutations per generation, and he just basically does multiplication to figure out the time to most recent common ancestor. And it's not like more sophisticated than that. Now that's a problem, but we're not going to like make a thing of that right now. We're going to talk about how he actually got those numbers. So take it away, Dr. Mays. What in the world?
For people who haven't read the book, it's the entire premise of it is he takes sequence data, which he never produces any sequence data himself. He always gets it from other sources. I don't think Ken Ham is generous enough to buy him an actual lab where he can do work. So he pulls sequence data from other sources and he builds trees using very rudimentary kind of approaches. And then he sort of rationalizes the science around those trees to get them to say what he wants, what he believes, right? And so for this book, he wants to say that human Y chromosome variation can be traced back to the 3 sons of Noah that left the ark, 4,500 years ago or so. Okay, so that's the entire premise. So it's, most people know, for males have an X and a Y chromosome and females have an X and an X. And so he's using sequence data from the Y chromosome, which is only in males and only passed from fathers to sons, to do this sort of patrilineal history that he says he can point to on the tree. This branch is from Shem. This branch is from Ham. This branch is from Japheth or whatever. And those are it proves that Noah's Ark was real and everyone's descended from those children of Noah. Right? So that's the premise of it. And so to do that, he has to do 2 things. He has to generate a tree and root it. And I'll talk in a second about what that is. And then he has to have a mutation rate, which is, like you said, he has to count the number of changes that take place over time and kind of map them on this tree, right, to get his time frame. This is this molecular clock thing. Now, no evolutionary biologist is as firm a believer in a molecular clock as Jeanson is. Like, for him, it's gospel, basically, the way he does it. Real evolutionary biologists, including myself, when we use mutation rates and genetic trees to infer things about the past, we give ourselves a lot of wide latitude in that because they're not very accurate to do these things. And the idea that you could use a molecular clock to get down to a few decades is just, for something that has generation times like a human, is just silly, really. But this is what he does. So the root the tree and then generate the mutation rate. So how does he generate the mutation rate? So he looked at a paper. I'm going to share my screen now.
Got you ready.
Let me just... Okay, so if you can see that tree there. So this is a tree from one of the papers that he cites where he says he derived his mutation rates. Now, this paper was done to look at what we call structural variants in Y chromosomes. Structural variants would be like a chunk that's missing or a chunk that gets added or something like that, right? And the purpose of this was not to look at what we call single nucleotide variants. That is a change from an A to a G or a T to a G or something like that, right? They didn't have the resolution to really do that. But Jeanson ignored that, and he sort of treated this like, well, they said that they were just going to study the structural variants, but buried in this paper are the single nucleotide variants. And he always sort of alludes to this fact like we're hiding something. Like really deep down, we know that the data says the human history is only a few thousand years, and we just sort of flub the results and hide it, which is silly. And so he believes himself as capable of digging through these papers and finding what the real numbers are. So how did he do that? So he looked at this tree, and if you look at the tips of the tree, there's, I think, 17 of these little forks at the end, right? And you can see that they're little branches that are really short, and they're at the very, very tips. And he looked at those and says, because this tree is made from sort of data where you had triads, you had a mom, a dad, and a kid all the time, right? And so he said that those tips of the branches are fathers and sons, since this is a Y chromosome tree. And I thought, is that true? It's like, did they make this just from the fathers in the study, or did they? And so I emailed one of the authors, emailed a couple, and they got back to me like really quick, which very often doesn't happen. They got back to me really quick, and I kind of embarrassing. It's embarrassing to talk to your colleagues about some of this stuff unless that they're already into it.
I can just aside, can confirm.
Yeah.
Can confirm. If you talk to people in evolutionary biology about young Earth creationism and they don't follow it, they're going to look at you like you have three heads.
Yeah.
That is true.
It's like talking to it's like saying you're trying to debunk a flat earther. Why would you waste your time on this? But this guy, to their credit, they were really helpful. So I emailed him, and I just asked, are the tips of those trees, are they fathers and sons? And he said, oh, yeah, they are. Oh, okay, well, that's right. And I kind of told him what this guy was trying to do with his data, and people aren't really into debunking creationists very often, but when you tell them that their paper is being used for some pseudoscience thing, they tend to want to
deal with it.
So he said, well, yeah, those little short forks at the ends of the tree, there should be 17 of them, I think. You can count them on the screen here if you want to. Those are fathers and sons. And so Jeanson looks at those and says, okay, well, the difference in the branch lengths, because the length of the branch is this is a distance space tree, which for people who don't know what that is, if you take all the sequences and make a table, and so you have the sequences listed on one axis of the table and the other axis, in each square intersecting cell or square in that table, you would write in what's the difference between that sequence and the other sequence. And so you would get this big table of all these pairwise distances, is what we call them. It's just the number of differences between that sequence and another. And then you can take that table and use an algorithm to say that, okay, the ones with the fewest differences have shorter branches, the ones with the bigger differences have longer branches, and you can build a tree. All it is a distance space. It's just telling you the diversity of those sequences on there in terms of their pairwise distances from one another, right? So that's what this tree is. And so Jeanson looked at this and looked at those branches on the tips and said, well, the branch length there should be the difference between a son and a father. And how do you get differences between a son and a father? Well, mutation. And so he measured that, and I'm not kidding about this. He measured that by taking a screenshot, just like I did here, of this tree from the PDF of the paper online. He took a screenshot and he blew it up, and he took the little marker at the bottom, the scale marker that says 50 mutations, and he used that as a ruler. And he measured the branch lengths, these really short little itty bitty branch lengths, at the 17 little bifurcations at the end of the tree, the tips of the tree. And then he said, that's the mutation rate between fathers and sons, which there's a lot of problems with that. The big one is, one big one is, this is like the weirdest, sloppiest, most inaccurate way to measure something. Like nobody goes and zooms in on a PDF and holds a ruler to the screen and measures something. It's just crazy. But that's what he did. And if you don't believe me, you can go Jeanson 2019, and I cited in the review that I wrote. That's in the links here, and you can go see for yourself. You can go to the paper, and it says exactly how he did it. The other thing, which one of those
before we
Go ahead.
move on, I just want to note in the link down there, it's all there. You could find like rigorously documented all this stuff. And also, I just want to note at this point, he didn't have to do this because like there are other papers that actually tried to figure that out
Yeah.
that he could have used. The
Yeah.
problem is the rate doesn't work for him. So
Yes.
this is how to get a number he wants. I just want to make that clear that it's not like nobody's ever done this correctly
And
and he's like trying to figure this out for the first time. Like this is out there. He just didn't cite the relevant work. Instead, he did this kind of half-assed thing to get the number he wanted.
in my correspondence with the author to this, one of the emails he sent me, he said, these are the mutation rates for the Y chromosome. And he listed like 10 citations on a little table that had the substitution, the single nucleotide substitution rates for Y chromosomes from like 10 different studies. And they're all between, I forget, like between 3 and 10 changes per like billion bases or something, tiny like that.
Yeah, times 10 to the minus whatever. There's very small numbers.
Minus 9 or minus 10 or something like that.
Just for people who are not intimately familiar with the calculations here. So these things are typically measured, if it's a mutation rate, it's typically measured in mutations per site per replication. So every time you copy, what's basically the probability of a base mutating. And then for substitutions, for fixed changes, it's changes per site per year, which removes generation time as a variable if you do it per year. So you end up, because you're talking about per site rather than per genome, you end up with a very small number, but you apply it across billions of nucleotides in a genome, you get like a countable number per generation is how it works out. But that's just for background for people who don't, haven't studied this in grad school.
Right. So that's how he measured this from a screenshot of a figure in a paper. Right. And the other thing I was going to note is I don't think he knows. I don't know how you could know which one of these branches is a father and a son. So he's saying that one branch in this little, bifurcation at the tip of the tree is a dad and one is a son, but you don't know which one. In some cases, they're the same length, right? In some cases, one's a lot longer than the other. You would expect the dad to have the ancestral base and the change to be in the son if he's measuring a mutation rate, but you don't know which is which. So he just measured that. Now, when I explained this to the author when we talked over email, and he said, well, because this is very complicated stuff. When you start, I usually sequence for my work like one gene at a time, like 800 base pairs at a time for like 12 genes to do some phylogenetics. That's pretty easy, right? To do that kind of thing. This sort of stuff is sort of high throughput sequencing where you have to get lots of little short reads from the same segment of the genome, and you have to stitch them together.
Mm-hmm. Yeah,
and when you do that,
overlap them.
Yeah, and so high throughput sequencing does a lot of sequencing, like the type of sequencing that I usually do for my work most of the time involves what's called Sanger sequencing. It's very, very accurate. So you don't have this problem of not being sure about what the base is, but you can only read a little bit at a time, like 800 base pairs at a time or so. High throughput sequencing, you have a lot more reads, but each read is less accurate. Now, Jeanson thinks that as long as you have coverage, he sort of ignores the rest of the inaccuracy of it. He says, well, he characterizes studies as high quality based on just how much coverage.
Based on the coverage, not the technique, right? Ignoring that there's like very specific biases depending on the sequencing technique that you have to account for. And he's just not even attempting to do that.
And what you're sequencing. So there's also problems with alignment if there's lots of repeats and things like that in what you're sequencing. So this is not a simple thing to either understand or to much less to do, right? And so my conversation with the author, he said, well, because we were only after the structural variants, we didn't worry about sort of the quality of the single nucleotide variants very much. So what that means is that in terms of the single nucleotide changes in this data, A to G, that kind of thing, right? I guess everybody knows what DNA has: an A, a G, a T, and a C, the bases. those single nucleotide difference, they weren't out to study that, so they didn't account for it. They weren't real careful about every single one, making sure it was the right base. So what he said was that this tree was made from only about 3,000 or so bases from the Y chromosome. This is not a tree from the entire Y chromosome, right? The error rate, he said, for the single nucleotide changes is about 1 in 1,000. So there's only 3,000 bases that make this tree, and the error rate's about 1 in 1,000. So you're starting to be very close to what Jeanson would think the mutation rate would be. So actually, what he said was, when I talked to the author, he said most of the differences between fathers and sons are errors and not mutations. And even for most of the structural variants they looked at, when they looked at structural variants in that paper, they find that fathers and sons are almost identical in every comparison for the other variants they looked at. So what Jeanson is measuring here are errors between fathers and sons, not mutations between fathers and sons. And that comes from the authors of the paper who said that.
And just a little more detail for everybody. If you look in the top left up there, it has known indels and novel indels on here, and it shows you along some of the longer branches how many of them are along each branch. Indels is short for insertion and deletion. So when we talk about structural mutations, we're talking about either you cut a piece out or you're inserting a piece. And often those insertions are tandem repeats where you have a piece and then it gets inserted immediately next to it. And those are the kinds of things where this sequencing has trouble because it reads in very short bits. So when you have a bunch of repeats, it's hard to do the alignment quite correctly computationally. But two, this is why the authors didn't care about the errors in their sequencing. They didn't care what specific bases were there. They were looking to see if there were any pieces that were added or any pieces that were removed. What the actual nucleotide sequence is secondary when your question is just what's being added or removed. So they didn't deal with those errors.
Yeah, the tree is based on, it's a distance tree based on single nucleotide variants.
Right.
But the differences between these long branches are much, much bigger than the error rate. So you could say, well, if they weren't worried about the single nucleotide variants, why did they build a tree from that data? Well, that's because you can build it. It's good enough to build a tree from, because the differences between the most of the branches on the tree is going to be much bigger than the error rate. The problem is when you start to draw inferences about branches that are really short, especially at the tips between fathers and sons, which are probably really zero. Any real differences due to mutations are going to be really close to what the error rate is. So that's why you can't measure. You can build a good tree from those data, but you can't measure mutation rates based on these little short distances between fathers and sons.
So let me make sure I'm clear on this, and also make sure we spell it out for the audience crystal clear here. The thing underlying Jeanson's work here is you can look at all of the diversity among human Y chromosomes, and you look at how fast human Y chromosomes mutate, and you could account for all of that diversity in approximately 4,500 years, according to Jeanson in Traced. But the data he used to calculate his mutation rate that's fast enough to let you do that is mostly sequencing errors, not actual differences due to mutations.
Yeah. And this is just coming from his own paper, if you want to call it that, his own article on the AIG website about how he did it. It's pretty clear. That's exactly what he did.
I was shocked at how not good the book was the first time I read it, and I didn't bother going into let me track down how he actually calculates his mutation rate. At some point, one of my notes in the book, as I was highlighting, I wrote to myself with the 3 mutations per generation, I said, probably wrong, but doesn't matter, is what I don't like. There's so many other problems. Like, we can put aside the fact that that was probably not calculated correctly, and I just didn't worry about it further. And then you came along and wrote this whole big long thing about exactly how it calculated it. And I figured it was going to be a little bit shady and not exactly how, like, you would do it for real.
Yeah.
But I was astonished to find that it involved blowing up a figure and using the scale bar as an actual ruler to count. It's just astonishing.
That's what he did. So yeah, and so that, it's like a lot of this stuff. Finding Darwin was kind of the same way. Like, he did not understand what the coalescent was, and yet he used that term in Finding Darwin, and he confuses it with a sort of distance measure between fixed differences between two branches on a tree. He confuses a coalescent approach with a molecular clock approach. There's overlap, and you can estimate sort of time with a coalescent approach, but counting up fixed neutral differences between two lineages and having a mutation rate there is not the same thing as doing the coalescent.
Not the same thing.
And so this is what happens is you point out that they don't understand something and they did something wrong, and then they just ignore it and go, okay, well, what about this?
Yeah.
But there's only two things you need to know for traced, and this is one of them. And if those things are wrong, then the rest of the book is just
doesn't matter.
Yeah.
So before we get to that second thing, I just want to point out that there was a recent piece by Dr. Rob Carter at CMI, Creation Ministries International. And in the comments of that article, people asked him about Traced and Jeanson, and it was really interesting and enlightening reading those comments, because he essentially said about the Jeanson strict molecular clock the exact same thing you just said. Which is really interesting, hearing that come from somebody who has used the pedigree-based mitochondrial rates to infer a recent mitochondrial Eve, right? But he comes out and says, like, that he didn't explicitly say that what Jeanson did was wrong or invalid, but he says that it involves assumptions that are not well supported, I think is how he said it. Yeah. Which I think is a diplomatic way of saying it.
The article you're talking about, and I found that kind of weird too. I think there's a lot of competition between these guys. I mean, they work basically for Christian nonprofit groups, and I was in the nonprofit world for a while, and money is a big thing in there, and so they're competing for donors, basically. So
yeah.
That's why they don't necessarily get along, I think, is why would they cooperate with one another and help the other's nonprofit?
It's a small pie.
Then it's just, I think that's all it is, really. But yeah.
So let's talk about the second problem here, which is how correct me if I'm wrong, but I think it's how Dr. Jeanson rooted his tree. And the way he actually set the origin and then how we went from there. Because like with coalescent theory, there's a lot of ways to do rooting. And like there's a lot of like valid ways to do rooting where you could pick a way of doing it and you can defend that technique, right? And there's different ways of doing it, and they have different constraints and different specific computational approaches, but like you can defend one way or the other when you pick it. And Jeanson doesn't really do any of that.
Yeah. So just to go over that here, let me share my whiteboard. Can you see my screen now?
You have a whiteboard on screen.
Okay, great. So how that works is, let's say you have a tree like this that's not a rooted tree. And let's say you have a tree with just 4 what we call taxa, 4 different things at the ends of the tree. We'll call them A, B, C, and D, right?
Warms my heart. To see this just warms my heart, because when I teach phylogenetics, I literally use this tree to talk about rooting. The simple 4 with the middle branch. This is how you do it.
Yeah. So this is an unrooted tree. Now, an unrooted tree has no historical information in it. And when I first saw Jeanson talk about these sorts of things, he would show what we call like a star tree, where there's sort of stuff emanating from the middle like this all around. And he would start to point to nodes on that tree to say that these are the sons of Noah. And I'm like, it's not a rooted tree. You can't say what came before the other. It has no what we call in systematics polarity to it, right? It's just showing the clustering of these things relative to one another. That's all it's showing. So just an example.
Before we go any further, I just want to direct everybody. I covered that in a different video. If anybody wants a lot of detail on it, if you're familiar with the famous 3-node trees for either the mitochondrial, the daughters-in-law, the Noah's sons' wives, or the Y chromosome tree where it's Noah's sons, and they have the tree with the 3 central nodes and all the branches. That's what we're talking about here. This star pattern unrooted tree. They use it all the time.
Yeah. So we can root this different ways. So if we root it on this branch in the middle here, we get this tree, right? Where A is more closely related to B and C is more closely related to D, right? If we root it on the A branch, then we get this tree where C and D are more closely related to other, and B is more related to C and D, and A is out here, right? If we root it on this branch, then we have B is here, and A, and you can stop me if I make a mistake, and C and D. All right. If we root it on, I'm just going to show all of them just for completeness sake.
Yeah.
C, then C is here. D is here, and A and B are on this branch. And then finally, if we root it on D, then we have this rooted tree with D here, C here, and A and B here, right? One tree, what we call topology, one type of tree with those taxa in those spots relative to one another, one arrangement, if you want to call it that, can have 5 different relationships depending on how you root it. So notice here in some of these things, C and D are more related to one another. In some of them, A and B are more related to one another. In some, C and D are more related to one another, right? So there's 5 different historical stories from one tree, right? That's just with 4 taxa. So imagine if you had hundreds, and so I forget how many are on Jeanson's tree, but there's dozens.
A
lot, right?
A lot.
There are hundreds of possible rooted, there's hundreds of possible ways to root that tree. He only tests 4 of them, right? 4. And he tests, the 4 that he tests are one that uses Neanderthals as an outgroup, what he calls the evolutionary way of doing it. And then the other ones are ones that he just basically eyeballs the tree and roots it such that it fits with the 3 sons of Noah. Those are the other 3 or so ways that he does this. So he only tests like 3 or 4 ways, the evolutionary way and the ways that are telling what he wants to hear, basically. So then he has those 4 different ways that he roots the tree. And then the question is, how does he tell which one is supported or good versus which one is bad? So he'll take each tree that he's rooted. So let's say you have a tree like this, say. I'm just drawing kind of a random tree here. Let's say this is your tree you're testing. It looks something like this, right? So what he says, and remember, this is a distance-based tree that he's rooted, right? So what he says, if this is time on this axis here, this is the past, is at the bottom of the tree, and the present is at the top, right? So what he says is, he says, okay, here there's one branch. Here at this time slice, there's 2. At this time slice, there's 3. This one, there's 4. Let's see where I'm at. This one, 1,
Yeah, you get
5, right? And this one, there's 1, 2, 3, 4, 5, 6, 7 at this one. Well, then he takes those, and this is a timeline of about 4,500 years, right? This is Noah. I can't write with my mouse. This is Noah, and this is now, right? And so this is the tree on this axis. So he says, well, there was, you have 1 branch, 2 branches, 3 branches, 4, 5, 6, 7, etc. Right? And so he would say, well, here at the beginning, there was 1 branch.
right.
The next 2, 3, and he draws this line like this: 5, and then 7.
Population, yeah.
Yeah, so he gets a curve like this, just counting the branches at each slice. Well, on the other axis, he puts population size. So this axis, the purple one, is the counting branches. The red axis here is population size. And its scale goes from like 0 to like 6 billion. I'm not kidding you. And he has this thing of here is there's basically no population growth, and then it shoots up, right? So what he does is he looks at these different trees, and he looks to see how they fit. I'll make this another color. So say this is another rooted tree. So maybe it looks like this, maybe his other rooted tree looks like this, stuff like that. And he just lines them up with this population curve. And he says the one that lines up the best must be true. And guess what? It just happens to be one that's not an evolutionary one, but one that fits with his paradigm. Now, what's the problem with that? The problem with that, there's so many, but you can't do this with a tree. You can't sort of count up the branches and say that this is the population now, this is the population has doubled. Now the population is 50% more again here. It doesn't work that way. You can't do that. And in fact, if he was using actual coalescent methods, they wouldn't look anything like this. If he was using actual coalescent methods, what a population that was exponentially growing, which is what he says is, happening in the past, and it's true. That's what's happening in the past, few hundred years. Not his timeline is off, but a tree from a population based on the coalescent that is growing exponentially looks like this. It has short branches at the bottom of the tree, and I'm drawing it kind of upside down. I did before, but you get the message. And it has really long branches here.
Right. It's to be clear, it's not exponential growth is not going to show you more branches. It's going to show you longer.
It affects the branch length.
It's the branch length, not the density of the branches on the tree.
And the reason for that is if you look at, say, this is a population of, I'll draw a bunch of, say, each dot here is a copy of a gene, right? And I'll draw a bunch of them. And then that's one generation. And these are copies of genes in the previous generation. Each dot is just a gene. It's just a stretch of DNA, right? And then so let's go back another generation here, right? I'm trying to make the exact same number, but I might make a mistake. But just
we get the idea.
You get the idea. It's the exact same number. Let's go back another generation, right? I'm going to do one more. So each one of these rows is a generation of copies of genes. Okay, so if you look at any two genes, let's look at, say, this one and this one, just picking two out of there. What's the chance that those two genes came from the same copy in the previous generation? Well, it would be 1 out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I have 12 here. So that's not too likely, right? So maybe this one came from this copy and this one came from this one. Well, what's the chance that these two genes came from a copy in the last generation? 1 out of 12, right? Well, that's probably also not very likely. Maybe this one came from this one. And so, maybe here and here, right? And maybe when you get back here, they coalesce. So here, this is the copy that they came from. And notice it's back in the past somewhere. Well, if the population is small, let's make the population smaller. 1, 2, 3, 4. 1, 2, 3, 4. Now, notice both of these populations are constant. They're not growing. They're not shrinking. They're the same size. I have 5 generations here. So if you look at 2 genes in the small population, let's look at this one and this one. What's the chance that they come from the same copy in the previous generation? 1 in 4, which is much more probable than 1 in 12, right? So they're going to coalesce much sooner. Maybe they don't do it here, but they might do it here. So their coalescent time is shorter than it is for the big population. That's why these branches are long, because the population in here, they're short because the population was small, right? This is what exponential growth is. It starts off really small for a while, and then it gets really big. That's why the branches are longer for the big population, when the population gets big, than for the small population. So if Jeanson were to look at this, use his method here, he would count this as 1, 2, 1, 2, 3, 4, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. And so what it would look like would be kind of like this, right? Where this is 1, 2, 3, 4, 5, 6 branches, and it would kind of level off at 6, 5, 4. It would increase a little bit at the beginning, but it would look like that. That's not at all like the curves that he's getting like this, that he says are exponential growth. If you applied his method to this, which is ridiculous. And then
it's just astonishing, right? That here's somebody who nobody's keeping how to do these techniques a secret from anybody.
No,
nobody's keeping this a secret. He knows you.
I know. I have a book right here that explains it all.
He could, he's talked to you before,
and
right? So he could have been like, "Hey, Herman, you want to just check this for me? Just make sure this is cool with how this works." He could have someone could have looked this over and caught this error, which seems to be something that anybody who does this regularly would be able to catch.
Of course. Yeah. So here's another one. So one thing he does too is he does this trick of sometimes when a branch is too long, like for instance, he talks about in the book, a branch in Africa is much longer than what it should be, based on his idea that there's these 3 sons of Noah and the way that he's rooted the tree. It's too long. And he just hand waves and says, well, that's probably because the mutation rate in that population was faster. Well, okay. He just throws it out there just to explain it away.
Why not? Sure. Why not?
Yeah. And then another time when he sees a tree like this, that's long and then has a lot of short branches at the top. And he does this, he talks about this in the context of Native Americans, right?
Right.
And he says, well, the reason that tree is like that it has one big long branch, is because there are, and it's a funny thing because he's getting sort of close there. He says there are these branches here that were extinct, right? And if he could still see those branches, then he could count across like this, and he would get his curve.
You get the right size. Yeah.
You get the right size according to his weird way of measuring, getting population size from a tree, right? Well, those populations go through bottlenecks. And what a bottleneck looks like is this. So if you have a tree, say like this here, I'm drawing it, it doesn't matter how you draw trees. You can draw them upside down a lot of times here. Let's say you have a tree like this with sort of more or less the branch points are kind of spread out a little bit, right? They're not all bunched up one way or the other. And this is for a constant population size. You would get a tree like this. Now, what happens when a population size goes down? Like it crashes and it stays low for a while, and then it goes back up. It might not go back up all the way to where it was, but then it steadies out again. So we call that a bottleneck. We call it a bottleneck because it looks like a bottleneck. The population is big and it gets smaller and then it expands. It's not just that it gets smaller, but it gets smaller and expands. That's what we call it a bottleneck. What does the tree look like on the other side of that? If the bottleneck is really severe, it looks like this. Exactly what the trees he is talking about look like. As it goes on, those branches get longer. And so over time, it starts to look more like this. Like that. Those little short things here are exactly because of this, a small population. They happen during the bottleneck.
And then as the bottleneck recedes into the historical past, the nodes that are the result of the bottleneck move.
The branches get longer because the population is bigger now. And it starts to look more like that. So the coalescent times are longer.
Yeah.
But he just makes up all this. I don't know if I can say this on your channel, but bullshit.
Yeah, you can say it. It's fine.
There's not a better word for it. It's such a useful word in the English language, actually, that he makes up all this bullshit to explain all this stuff when, like you said, it's all out there already. And he just doesn't bother to look at any of it.
Yeah. That's gene genealogies, variation and evolution. There are books on this that anybody can go and find and look up. You can do this for real. This is so my expertise is not in doing these exact techniques in grad school. My work was adjacent to doing these. For Dr. Mays, for just so you know, my lab was all viral phylogenetics and evolution. I was working on like mutation rate stuff, so I was upstream of. So I was like figuring out like what the substitution matrix should look like while my colleagues were doing the actual phylogenetic stuff. And like I, as somebody who was like worked with people who would do for anyone familiar with the program Beast, like work with people who were using Beast all the time, like that's that one degree removed there you go. Right. So like 1 degree removed from actually doing this is where my expertise lies. And I can read this stuff, be like, this is not how any of this works. This is not how you do these techniques. This is not appropriate. And then for somebody who actually does do this stuff, I can imagine just your head exploding as you're digging into these techniques.
This idea that he can look at a distance tree, and just go across each thing and say, this is how big the population is here. This is how big it is here. This is how big it is here. That's not how that works.
Yeah. Using a number of branches.
What he doesn't understand is you would get a branching tree of sequences, even if the population were constant over time. He thinks that you only get branches if the population is growing exponentially. If it's the same size throughout time, he thinks there's no branches.
There's no branches.
It makes no sense,
which is completely contradictory with his per generation mutation rate molecular clock. There's still reproduction happening. Yeah. There's still mutations happening. There have to be branch points.
Exactly.
Even if you're like,
it's weird. It's weird. And he can do it. And this is the point of all this. Like, we can go forever, and people do this, and I do it. We can go forever and and tear apart the science of this and explain why it's wrong. One mistake in that is, in a way, it means we're taking him seriously.
Right. And
Right?
that's not the point. The point isn't that the science is valid.
And it's just, for him, he can say all this stuff because we are not his audience.
Yeah.
His audience is a group of sort of culture war, evangelical, fundamentalist Christians who mistrust science and scientists, and who in some ways want the United States in particular to operate as a de facto theocracy. That's his audience. They are not going to dig deep on coalescent approaches or distance approaches to building trees from molecular sequence data. They don't care. All they care about is this telling me what I want to hear? It's confirmation bias on his part and his audience's part. They just care that it's telling them what they want to hear, and it's coming off as sounding scientific. And that's what he does. But if anyone who knows what they're doing digs into it. Now, if you don't, this is all really complicated stuff. And if you don't have time to dig into it, or you don't understand all this stuff, but you're wondering, is this guy credible or not? There's just one thing to remember. If he's credible, why aren't these astounding findings in the pages of Science and Nature, and not on AIG's website, basically the equivalent of that? That should tell you everything you want to know about credibility. It's because he can't compete in that arena, and he'll never publish this stuff there, because once anyone who knows what they're doing takes a look at this, they realize that it's bullshit. That's it. and the rest of it is just him sort of making this seemingly scientific argument, because ultimately what he wants and what his readers want is basically a sort of theocratic imposition into secular society. They want this stuff taught in schools. They want the religion to be rubber stamped as sort of official. They want to outlaw gay marriage. They want to erode rights for LGBT people. That's the agenda here. And yeah. I think we get very lost in debunking the science, and we forget why they're doing this in the first place. And that's why.
Yeah. I think that is a perfect summary. I don't have anything to add to that. That is exactly the right approach to this. Just remember, science is not the audience. People who are concerned with the science, not the audience. This is for people already on board to give some kind of plausible justification. But it's not, like you said, it's never going to appear in Science or Nature, because that's not the goal. That's besides the point. So
he knows that.
Yeah. So everybody, this has been Dr. Herman Mays talking about the methodology from Dr. Nathaniel Jeanson in Traced. This has been a lot of fun. I hope everybody enjoyed that. And if your jaw is on the floor, please pick it up before you go about the rest of your day, everybody. So Dr. Mays, thank you so much. Oh, one more thing before we go. Just so the two books that you showed, in addition to Traced, you had there was the Gene Evolution one and the Beast one. Just want to make sure people see those, because
one is Alexi Drummond's, who just sort of made Beast.
There we go. So Bayesian evolutionary analysis with Beast. There you go. And then the other one.
Jeanson was actually interested in using molecular data to infer population history. You would use a coalescent approach to do that. And that usually means likelihood and Bayesian methods like this.
Yep.
Right? And so the coalescent stuff is explained.
There you go.
This, which is Jotunheim's book on coalescent theory.
Again, not keeping this secret, everybody. Nobody's keeping this stuff secret.
No.
Anybody can look this stuff up. It's all there.
Like 10 years old.
Yeah.
I mean,
all right.
Yeah.
Well, thank you for watching, everybody. I really appreciate, it's the middle of the day, so I appreciate everyone who's here watching. Dr. Mays, thank you for taking the time to do this. I really appreciate it. Everybody, if you haven't already, hit the little like button. Do that unless you're really mad right now, in which case hit the dislike button. That's fine too. I don't really care. Either way, whatever floats your boat. We are going to call it a day. thanks everybody. I don't have anything to say is coming up because I don't know what's coming up next, but I do have some stuff that'll be coming up soon. So this has been Dr. Mays talking about Jeanson's methodology in Traced. I hope everyone feels enlightened and flabbergasted. Have a great day, everybody.
Thanks.